Within a sealed 5-gallon plastic receptacle positioned on your back patio, resting alongside a fractured terracotta pot and a sack of pine bark mulch, something truly remarkable is transpiring at this very moment. Every 48 hours, the living population contained within that bucket experiences a doubling, and the substance that these minuscule lives are generating, for the cost of a single organic potato and a tablespoon of sea salt, possesses a molecular structure identical to a category of plant growth enhancers that the United States Department of Agriculture dedicated 40 years and over 500 pounds of bee-gathered pollen to isolating. That statement may seem like an overstatement. By the conclusion of this presentation, you will comprehend that, if anything, it is an understatement. Because the reality of what is occurring within that humble container surpasses the scope of this video's title. The agricultural innovators from Korea who conceived of this approach did not merely stumble upon an economical plant nutrient. They reverse-engineered a 400-million-year-old dialogue between forest microorganisms and plant roots. A dialogue that activates nearly 1,000 genetic pathways within your tomato plants, your rose bushes, your blueberry plants, and your raised garden beds, the instant those microbes make contact with the soil. In the ensuing 18 minutes, you will depart with the precise methodology, the rules for dilution, the scientific explanation for its efficacy at a molecular level, and the resolution to the singular query that occupied American researchers for 4 decades. Please be seated. Pour yourself a warm beverage. This information warrants your complete attention. 1. The 1979 crystal that revolutionized plant science. Let me commence with a narrative that very few individuals outside of the botanical research community have ever encountered. In 1979, within the confines of a tranquil laboratory in Beltsville, Maryland, a collective of plant chemists affiliated with the United States Department of Agriculture finally obtained 4 milligrams of a minuscule crystal. 4 milligrams, that is less than the mass of a single grain of rice. To extract those 4 milligrams, they had processed more than 500 pounds, equivalent to 227 kilograms of rapeseed pollen, meticulously gathered by honeybees from vast agricultural expanses. The endeavor spanned the greater part of a decade. It necessitated pilot-scale solvent extraction processes, the collaboration of multiple specialized USDA laboratories, and a caliber of patient industrial chemistry that is simply no longer adequately financed. When that crystal finally achieved sufficient purity for examination under X-ray crystallography, the scientific community collectively held its breath for a moment. The substance they had isolated was a steroid, a genuine steroidal hormone, possessing the same foundational molecular structure found in the steroidal hormones circulating within your own physiology. For 2.5 centuries of contemporary botanical study, it had not been conceived that plants produced actual steroids. The team published their findings in the esteemed journal Nature in that same year and christened the compound brasinolide. Presently, brasinolide and its related compounds, collectively known as brasinosteroids, are acknowledged as the 6th primary classification of plant hormones. They are featured in every current plant biology textbook alongside auxin, gibberellin, cytokinin, ethylene, and abscisic acid. The trade publications of that era bestowed upon brasinolide a memorable moniker that persisted: the mastermind steroid. You are about to discover the rationale behind this designation. In 2010, 2 scientists from the Carnegie Institution for Science, Dr. Yu Sun and Dr. Ji-Young Wang, presented a groundbreaking research paper in the journal Developmental Cell. They meticulously mapped, for the first time in recorded history, the precise function of brasinosteroid within a plant cell. The revelation was astonishing. They identified a master regulatory protein designated BZR1, which, upon activation by brasinosteroid, directly governs the activity of 953 distinct genes within the plant's genetic makeup. Pause and absorb the significance of that number. Nearly 1,000 genes. Genes responsible for regulating cell elongation. Genes that oversee the transport of water within the plant's vascular system. Genes that command the plant's immune responses against fungal adversaries. Genes that determine whether a tomato will succumb to cracking under heat stress or endure a sudden frost. The keys to virtually every aspect of plant well-being, all governed by a single hormonal signal. And the promise of this video is that you are about to cultivate that signal in virtually limitless quantities within a bucket on your own porch. That assurance is not mere marketing, it is a statement of microbiological fact. 2. The Korean farmer who perceived what the USDA overlooked. Now, allow me to transport you 10,000 miles from that Maryland laboratory to a small mountain village in South Korea during the 1960s. A young farmer named Cho Han-kyu has just received a glossy promotional brochure from a representative of a Western chemical enterprise. The brochure purported miraculous crop yields if Cho transitioned to their novel synthetic fertilizers. Many of his neighbors complied. For 2 growing seasons, their harvests surged. The rice panicles were heavier than the village elders had witnessed in a generation. Then, by the 5th year, the soil that had sustained those families for centuries began to deteriorate. The earthworms vanished. The scent of damp spring earth, a fragrance familiar to every farmer's child, disappeared. Plants weakened. The incidence of disease escalated. Each subsequent year, the farmers were compelled to purchase additional chemicals merely to maintain the yields they had once received gratuitously. Cho Han-kyu refused to participate in this cycle. Instead, he ventured into the untouched forest bordering his village. He knelt, gently pushed aside the uppermost layer of decaying leaves, and gathered a handful of the dark, friable soil beneath. He held it in his palm, contemplating it for an extended period. No human had ever administered a single drop of synthetic fertilizer to that forest floor. Yet the colossal oak and pine trees had stood there for centuries, cultivating their own fertility from nothing more than rainfall, sunlight, fallen foliage, and the patient activity of unseen organisms within the soil. He posed the question that altered the course of his life, and ultimately ours. If the forest can sustain itself without cost, why cannot a farm? For the subsequent 40 years, Cho Han-kyu and his son, Dr. Cho Young-sang, systematically explored methods to harness that invisible forest ecosystem and integrate it into cultivated agricultural fields. In 2002, they established the Jadam Institute. Jadam, meaning in Korean, people who resemble nature. According to Dr. Cho's own published statistics, conventional chemical farming incurs expenses of approximately $5,000 per acre annually for an American producer. The complete Jadam system, utilizing ingredients you likely already possess in your kitchen and from your local wooded areas, reduces that cost to around $100 per acre. Re-examine that comparison, $5,000 versus $100, yielding results that peer-reviewed Korean and American researchers now describe as equivalent to, or in certain categories superior to, conventional crop yields. The pinnacle of this entire methodology is the recipe currently residing on your back porch. It bears a simple Korean designation, Jadam Microbial Solution. Most farmers simply refer to it as JMS. 3. Why doubles every 48 hours is, in fact, an understatement. Let me address the title of this video directly. Some of you may have clicked due to the phrase doubles every 48 hours, appearing excessively dramatic to be credible. Here is the scientifically accurate response. That phrase is not sufficiently dramatic. A healthy soil bacterium, and we will identify the specific types momentarily, exhibits a doubling time under optimal circumstances ranging from 20 minutes to 2 hours. That is not an exaggeration. It is textbook microbiology. A solitary bacillus cell introduced into a suitable liquid medium with the appropriate nourishment will replicate into 2 cells in approximately 20 to 30 minutes. 2 become 4. 4 become 8. By the 10th hour, a single cell has theoretically produced over 1 billion progeny. Naturally, no receptacle on Earth can replicate those ideal laboratory conditions, and the rate of multiplication slows as resources become constrained. However, the overarching principle remains. When you introduce a handful of forest leaf mold containing somewhere between 1 and 10 billion living microorganisms into a bucket of mineral-rich water supplemented with a boiled potato as sustenance, you are initiating an exponential growth trajectory. The total microbial population within your bucket does not merely double every 48 hours, it proliferates explosively. The visible foamy layer that forms on the surface by the morning of the third day is the tangible manifestation of that surge. And here is the crucial aspect that the title does not have the space to convey. Among the species present in that burgeoning population are members of 3 particular genera that have been extensively studied in peer-reviewed microbiology over the past 20 years. These are Bacillus, Pseudomonas, and Azospirillum. These 3 groups, along with several others, have been confirmed in numerous published articles in journals such as Frontiers in Plant Science, Plant and Soil, and the National Library of Medicine's PubMed database, to actively synthesize and secrete plant growth hormones as an integral part of their routine metabolic processes. Auxins, gibberellins, cytokinins, abscisic acid, 4 of the 6 principal classes of plant hormones are produced freely and continuously by the bacteria multiplying within your bucket. Hundreds of research papers attest to this. The science is not a matter of contention. Forest bacteria function as pharmacies. They produce medicinal compounds for plants, and every 48 hours, that pharmacy doubles in scale. 4. The concealed dialogue that activates your plant's steroid production. Now, I wish to introduce an even more remarkable aspect that virtually no gardening channel has explored yet. Because the title of this video promises you an abundance of plant steroids, and the source of these steroids is more sophisticated than direct bacterial synthesis. Recent peer-reviewed research, including a 2024 study published on Science Direct, examining a soil bacterium known as Bacillus endophyticus strain J13, has revealed something extraordinary. These microorganisms do not simply deliver pre-formulated hormones to your plants as a courier might drop off a package. Instead, they transmit chemical signals through the root zone, and those signals trigger the plant's own internal brassinosteroid production machinery. The plant enhances its internal synthesis of those mastermind steroids in response to a microbial exchange occurring at its root tips. Allow that concept to settle for a moment, as it fundamentally alters the perspective. The bucket on your porch is not merely a fertilizer dispenser. It is a catalyst for awakening. You are not introducing artificial chemicals to your plants. You are not even adding hormones directly. You are introducing a population of microscopic pharmacists who, through a communication system predating dinosaurs, will gently tap upon your tomato plant's molecular doorstep and remind it how to activate the 953 genes it already possesses, but which have been suppressed under the chemical regimen of conventional cultivation. The plant undertakes the task of generating its own steroids. The microbes serve as the signaling mechanism. You, situated in your backyard with a 5-gallon bucket, are the host who has invited them all to partake. And because you can prepare a new bucket each week, each month, each season for the remainder of your gardening endeavors, the supply of these steroid-signaling microbes is, in practical terms, inexhaustible. The title is not a marketing ploy. It is an accurate depiction of how this biological process functions. Nature has been operating this precise biochemical system in undisturbed forests for 400 million years, long before the advent of any chemical corporations. Master Cho was the first to discover how to containerize it, and the recipe is so straightforward that I am about to guide you through its complete execution. Free Bucket Blueprint, your take-home companion. Before we delve into the recipe itself, and I intend to present the recipe with absolute precision, including every pertinent detail, please pause for 30 seconds so you can depart from this video with something tangible in your possession. On your screen at this moment, you will observe a QR code. Open the camera application on your smartphone, position it over the code, and tap the link that appears. That link will immediately initiate the download of a free PDF titled The Jadam Bucket Blueprint. Within this document, you will discover the master dilution chart for every plant variety in your garden, a month-by-month application calendar tailored to your specific American growing zone, a troubleshooting guide for addressing any issues such as an unusual odor from your bucket or a failure of the foam to form, a printable checklist of the 5 essential ingredients, and a quick reference card that you can affix to the interior of your garage door, thereby eliminating the need to recall any numerical data. This blueprint is entirely complimentary. No email registration is required. There is no hidden agenda. It is yours to keep, to print, and to share with any fellow gardening enthusiast who could benefit from this knowledge. If you are viewing this on a smaller display or have your hands occupied at present, the identical link is prominently featured at the very apex of the description box below this video. Please take 10 seconds to retrieve the blueprint and rejoin me here, because what follows is the segment where we will construct the bucket together. 5. The 5-ingredient recipe that costs